majestic, enigmatic creatures are actually essential wardens of our marine environment. Found in every ocean across the world, they play a fundamental role in ensuring balance and stability in our oceans. Sadly, this role is jeopardized by overfishing and habitat loss. Around 3 billion people rely on fish as their primary source of protein. Ongoing declines in shark populations could have devastating and unpredictable impacts on marine food webs, and even our own food supply. The plight of sharks and rays was the spark for the Save Our Seas Foundation, founded over 15 years ago. Through the funding and support of research, conservation, and education initiatives, we are dedicated to safeguarding the future of sharks and rays. Today we have 3 centers based in Seychelles, South Africa, and the USA. Since 2003, we have funded over 300 projects on over 60 species in over 50 countries. We have also partnered long-term with several projects, including the Shark Spotters in South Africa, the Shark Lab in the Bahamas, and the Manta Trust. We're passionate about our mission: protect the future of sharks and rays and our oceans for generations to come. We can and will make a difference. The Save Our Seas Foundation.
I used to actually be terrified in any water where I couldn't see the bottom. Tales of monsters lurking in the deep. What are these enigmatic, crazy creatures? The first recognizable ancestors of sharks existed before trees, whereas now they could be facing collapse in a single generation. I actually think it's very hard to know just how much you love something until you lose it. Grief is the price we pay for love. The thing that drove me on this path was a pure passion for the animals themselves. I didn't start out wanting to save them because I didn't know that they needed saving or help. This one's for the ocean. I realized that if we want to help save sharks, we need to make sure they have safe places to live in.
Sharks and rays, they are ancient, enigmatic, misunderstood, and essential to our oceans. But these important animals are under immense pressure. As the Save Our Seas Foundation, we're here to help. Through supporting research, conservation, and education, we work to ensure a future for sharks and rays. The Save Our Seas Foundation. Together, we can make a difference.
Hello, everyone. Welcome to tonight's Save Our Seas Foundation Distinguished Speaker Series. We're so happy to have you. My name is Lance. I am the Director of Sustainability at the Museum of Discovery and Science in beautiful Fort Lauderdale, Florida. Tonight we're bringing you another really amazing speaker. Dr. Ramón Bonfil will be here to talk to you all about his amazing shark research. And I'll introduce him in 1 second. But first, I just want to say thank you to the Save Our Seas Foundation for making this whole series possible. This is the fourth of this year's Distinguished Speaker Series, the fourth installment. We have 3 previous years you guys can go check out if you're interested on the Museum of Discovery and Science YouTube channel. You can check out 3 years worth of talks and find out more all about shark research, conservation, education, and all the amazing things that the Save Our Seas Foundation is doing. If you ever find yourself in sunny South Florida, we would love to have you at our museum. Here's a lovely picture of us here. Our mission at the Museum of Discovery and Science is to connect people to inspiring science. We're going to strive to do that to you with you tonight by sharing some really inspiring science, shark research. Everything we do is driven by four strategic educational pillars: physical science education, early childhood education, health and wellness education, and then tonight is that environmental sustainability, where we're talking about ocean conservation. So, like I said, if you ever find yourself in Fort Lauderdale, please come on by and take a visit. Tonight, like I was saying, we are bringing you another amazing speaker. Dr. Ramón Bonfil is an internationally recognized elasmobranch researcher with over 35 years of experience in shark and ray conservation. He's worked in fisheries management and marine ecology as well, with a Master's of Science from the University of Bangor in the UK, and a PhD from the University of British Columbia in Canada. His career spans work across 5 continents, including working with the Wildlife Conservation Society and the federal universities in Brazil. He is a founding member of the IUCN Shark Specialist Group and currently serves as the National Research Chair at EcoSur in Mexico. Dr. Bonfil's research has led to groundbreaking discoveries in the ecology of great white sharks and has been instrumental in global conservation efforts, including securing CITES protection for several threatened species. He has published extensively with over 8,000 citations of his work. Now based in the Mexican Caribbean, Dr. Bonfil leads conservation projects in coastal lagoons and biosphere reserves, using cutting-edge tools like satellite telemetry and environmental DNA to study manta ray migrations and to locate and protect last remaining sawfish in Mexico, as well as inspiring future marine conservationists and mentoring the next generation of ocean stewards. So, Dr. Bonfil, we're so happy to have you here tonight. Welcome.
Thank you very much, Lance, for your very kind introduction, and hello to everyone. Should we begin?
Sorry again?
Should I begin or
Yeah, sure. Please. Yeah. So go ahead and share your screen. While you're doing that, I'll just kind of do a few little things for the audience. You guys are free to ask questions tonight. Ask questions. You can do that through the chat. Please put any questions you have there. I'll be monitoring the chat for everyone. We will have some dedicated question and answer session at the end, but don't wait to the end to ask your questions. If you have something that's really awesome question about what we're talking about at the moment, I'll chime in and ask that for you. Otherwise, I'll just wait to the end. So feel free to put questions into the chat as we go along. All right. With that,
all right.
Take it away. Yeah.
Okay. Thanks a lot, Lance. All right. Today I am gonna tell you a little bit about the latest research that I've been doing here in my native country, Mexico. I was originally from here, and then I traveled extensively throughout the world. I lived in many countries for 25 years, and I decided to return to Mexico to finish my career in marine science and shark conservation in EcoSur in the southern part of the Quintana Roo state, in the same state where Cancun and Playa del Carmen are. We're a little bit to the south of that. And I'm going to tell you precisely about what we've been doing in the last few years here in the Mexican Caribbean. To begin with, I'm gonna put a little bit of context. I guess most of you are familiar with what sharks and rays are. They are called elasmobranchs. They're a group of fish that is characterized by having a skeleton that is made entirely of cartilage. And there are a few other characteristics that separate them from bony fishes, but I'm not gonna get into that. The point I want to make is that the diversity of sharks and rays is really, really great. There's all types of forms, shapes, colors, sizes in sharks and rays. And among them, we have the largest fish that lives in the ocean with the whale shark right here, where the pointer is moving on the top right. And just below it is the smallest shark that we know of, the cigar shark. And these are just to exemplify. One of them is no more than maybe 12, 15 inches. And the other one can measure up to 18 meters, they say, 15 at least, for sure. So that shows you the wide range of sizes they can attain, and they occupy all kinds of environments. Now, the difference between sharks and rays is basically that rays are like sharks that were run over by a big truck, and they are flat. It's like a pancake shark. But other characteristics of their life history, biology, ecology are very, very similar. And in some cases, it's very easy to distinguish a shark from a ray. If it is flat and has the gill slits on the underside, on the ventral side, it's going to be a ray. And if it is like a torpedo in shape and has the gill slits on the sides, it's going to be a shark. But there are a few forms of sharks and rays that are kind of difficult to understand and to distinguish. For example, this one here has half of the body like a ray and the other half like a shark. And many people don't understand what they are. And this other one here also has features that resemble rays and others that resemble sharks. But basically, these two are in fact rays, as well as the sawfish, which is this one right down here with a very long rostrum that has teeth all around the sides. Those are rays. But this one that we have here that looks like a ray, it's a shark. It's a wobbegong or carpet shark. So this is just to show you that sometimes the relationship between sharks and rays is so close, they are sister groups, that in some species, there are still remnants of that shared ancestor or convergence in evolution that makes them look very similar to each other. Now, an important part of sharks and rays is the biology they have. And in particular, the reproduction of sharks and rays is characterized by having internal fertilization. They mate, and here we have two images of a couple of sharks mating. In the upper left, it's the moment where the male is trying to grab the fin of the female to start the mating. And in the bottom one, you can see the two of them already mating to perform the fertilization of the eggs by the sperm of the male. And the same happens with rays. Here we have two manta rays that are beginning the courtship, and the male is again trying to bite the pectoral fin of the female in order to mate. And in the bottom one, they're already mating. So all sharks and rays mate and have internal fertilization. Then the development of the babies happens in many very different modes. There are a wide range of strategies, what is called oviparity, oophagy, and there are viviparous sharks, and rays, many modes of viviparity. And I will explain in a minute what exactly viviparity means. But what I wanted to make you notice is that most of the species of sharks and rays are viviparous. They have their young are born from the mother, just like you and me and dogs and cats. they have to develop inside the belly of the mother, and then they are born alive into the environment. A few species of sharks and a few more of rays have oviparity. They lay eggs. So the fertilization is inside the female, but then she goes like a chicken or like a bird and lays eggs in the environment and leaves them alone, and they develop on their own. But this is really the lesser amount of the species. Most of the species of sharks and rays have some sort of form of viviparity. They are born alive. And this is part of the biological constraints they have. So this is viviparity. This is a great shot from lemon sharks, probably in Bimini Lagoon in the Bahamas. And you can see a baby that is already born and is trying to break the tie between the placenta and the umbilical cord. And there's another one that is halfway out of the cloaca of the female. These sharks are born in very shallow waters in tropical lagoons around mangrove forests, in islands and coastal lagoons. And this is viviparity, and it's very important. It has a lot of implications for what is happening with sharks and rays worldwide. As I explained, this is another shot that I took in the Red Sea many years ago of a sharp-nosed shark that is being opened. And you can see here the stomach, and then you can see the parts of the liver, and then the uteri. The females have 2 uteri where the embryos develop. In the next figure, you can see the uteri already outside of the female. This is the left uterus, and this is the right uterus. And inside of each of them, there are baby sharks being developed. And the final shot shows you all those little babies already here with one of them with the umbilical cord still attached, and on the other extreme, the placenta. Just like humans, some shark species develop a placenta that connects the tissue of the baby with the tissue of the uterus of the mother, and that's how energy and food supplies are passed from the mother to the babies, to the embryos, and that's how they develop. So at the end, there are many, many similarities between humans and these very, very ancient fish called the sharks. Now, one problem, as I mentioned, is that the size of the mother, the belly has livers and it has kidneys, the heart, it has all the organs that are vital for the survival of a shark. And on top of that, they have to share that space with the babies. That puts a strong limit on how many babies can a mother have. And that means that they have, in general, very low fecundity. They have very few pups every time they reproduce. And this is common to sharks and to rays. And another problem is of the reproduction of sharks and rays is that the pups take a very long time to develop, between 6 months and 22 months in some species. That's much more than in humans. We take 9 months approximately to develop and be born. Some sharks do more than that, even 22 months. Others less. But this is another important fact that we're going to have to take into account. Now, this graph shows you a little bit of how common it is to have very limited space for babies. This graph shows you for different shark species, about a dozen of them, what's the range of babies they can have. And you can see that most of the species are below 10. They can have less than 10 babies every time they reproduce. A few of them, like the tiger shark, can have up to 80, the blue shark up to 60, and the bramble shark up to 50 approximately. But in general, this is very, very limited compared to bony fishes that can reproduce, laying millions of eggs, each individual at a time. And this is very important. So the low fecundity of sharks is common to most of the sharks that we know about, both in Florida and here in Mexico, like the bull shark, the lemon shark, or the scalloped hammerhead shark. So that's very, very important. Now, the other problem we have is that the growth of sharks and rays is very slow. This graph shows you the length on the y-axis, the size of the shark, and on the x-axis, the age. And you can see that for these three species of sharks, this is the, I forgot for a moment. Well, this is the spinner shark in the center. Below is the sandbar shark, and this is the dusky, the dusky shark. So these three species of sharks should be also common to some of you because they occur in Florida as well as in Mexico, and they grow very slowly. These symbols, the stars, are placed here to show the age in which each of these species reaches sexual maturity. That is when they can start to reproduce. And this is around 11, 12, 9 years old. So it's almost like in humans. We cannot reproduce before we are 12, 14, 15. The same happens with sharks. And these two things, the slow growth and late maturity, means that they have very little power to replenish their populations. Sharks and rays are known as K-type selected organisms because of the slow growth they have and the high longevity. They live many, many years. They reach maturity, sexual maturity very late, 9, 10, or longer years. They have low fecundity, and also they have gestation cycles, the development of the babies that take a long time. All of this, when you add it up, means they have a very low reproductive potential. And this means that if you take half of the population of sharks in one go by fishing, it's gonna take them many years to go back to the same amount of sharks that there were in that population before you took half of them out. And that's the main problem we are seeing at the moment with shark fisheries worldwide and the conservation of sharks and rays. So elasmobranchs, sharks and rays, cannot sustain heavy fishing because they take a long time to replenish their populations. And this has been shown throughout history. This graph shows the subfin shark fishery in California in the 1930s, 40s of last century. And you can see that the graph shows that they were slightly fished, not very big, until the beginning of the World War II, and then the catches rose exponentially in only a couple of years because of the demand for vitamin A, which is taken from shark livers, or used to be taken from shark livers. And after a few years of fishing very heavily for sharks, this population went down dramatically in a very, very short time. This pattern of boom and bust in shark exploitation is common, is not unique to that particular population. Other parts of the world, other species have suffered the same fate. They are exploited very heavily, and soon after that, they start to go down. This is the porbeagle shark in the North Atlantic, around Canada, between Canada and Europe. And this was fished for meat, for human consumption in the 1960s, and the population rapidly went down to less than half in about 15 years. The regulations tried to help it to recover. There was a little bit of recovery, but they continued to fish them, not as heavily as before, but they continued to fish it, and the population seems to be going down. So this is common for all shark and ray species. Another problem we have is that the catches of sharks and rays in the world do not go down. People still need more and want more consumption of seafood and fishes. And because of that, the demand keeps growing. And this is a graph that shows the catches, the worldwide catches of sharks and rays from the 1950s to 2010. And you can see that it's continually growing. And when you put all this together, you can understand why sharks and rays have become recently, in the last 2 or 3 decades, an issue of conservation and a concern for the health of oceans. They are unfortunately, on the threat of extinction, the assessment of elasmobranch conservation status from 2014 made by the International Union for the Conservation of Nature Red List, of which I am part, found that 25% of the species of sharks and rays were threatened with extinction. 45% were without enough information to know if they were in trouble or not. And 24, because of that, because we didn't know what was going with all this gray area up here, if we extrapolate the numbers from the information we have, we can estimate that probably half of that is also in trouble. So it could be that in 2014, it was thought that maybe about half of the species of sharks and rays in the world were already threatened with extinction. That is, they were considered either vulnerable to extinction, endangered, or critically endangered. And these colors depict the different categories that we use at the IUCN Red List to classify species according to how well they're doing or how poorly we are doing with their conservation. So red is bad and yellow is okay. And gray is we don't know. So here you can see that almost 1/4 of them are either yellow, orange, or red. But the situation didn't improve. It is worsening after decades. Now, at present, over 1/3 of species are threatened, and this shows you a comparison of what we thought about the conservation of sharks and rays in 2014 versus what we know now in 2021. When more species have been assessed, the gray has diminished, but the line of threatened species has gone wider. Now there are more red, more yellow, and more orange species, and almost 37% of the sharks and rays of the world are thought to be in some trouble with possible extinction. So the situation is not particularly good. Now, what can we as humans do to protect and conserve shark and ray populations? There are many things that we can do. One is to set limits on the catches in fisheries by regulations of the governments. Say you cannot fish more than X amount of tons of sharks every year, but that is rarely done. Another is that regulations, other types of regulations of fisheries, for example, they can ban certain species or ban certain types of fishing gear that are particularly problematic, or either areas that cannot be fished. Prohibitions on species, fishing gear, and fishing areas are other possibilities. Also, we have the CITES appendix species. CITES is an international organization that deals with the international commerce of species that are in danger, being plants, animals, invertebrates, anything. So many species are already of sharks and rays are protected by CITES, and this means that to sell internationally products of sharks and rays, you have to demonstrate that you are not harming their populations in your country. And this is a way of helping sharks and rays. Another possibility is to establish marine protected areas that have no take, no fishing in them. There are very few in the world, but there are. And the other thing that I wanted to mention is a new tool that has been developed very recently, and it's called the Important Shark and Ray Areas. And these are areas that are studied by scientists and found to be of specific importance to a group, in this case, sharks and rays, because they are either nursery areas, for example, it could be special feeding areas for the species, or just areas where they aggregate, for example, to do something like mating. So we need to identify important shark and ray areas so that they can be used for more protection of sharks and rays. And in this context is that I'm going to talk about particularly about shark and ray nurseries. What are they? Okay, initially they were described many years ago very loosely, not very specifically, but very loosely, as shallow water areas where the juveniles occurred as opposed to adults, and where the newborns were born and remained there growing until they reached sexual maturity. But that was a very loose, very general definition. Later, other shark scientists proposed that there were two types of nursery areas, primary and secondary. The primary were those where the babies were born and remained during the first part of their lives. And then they moved to a secondary nursery area when they were a little bit larger, and they continued to grow in those areas. However, the problem with this definition is that they were not very good for going to a place and deciding, is this or is this not a nursery areas for this ray species or this shark species? So there was a little bit of trouble. Now, shark and ray nursery areas, I'm going to show you here, are protected from predators and feeding on whatever they can find in the area. Okay, so now, fortunately for us, a group of scientists realized what was the problem with the definition of shark and ray nursery areas. And almost 20 years ago, they did a scientific review of all the literature and thought about the problem, and they came up with a useful set of criteria to define nursery areas. And these criteria are three. Nursery areas are specific areas where the young less than 1 year of age are more abundant than in other areas, where they remained or returned to these areas over long periods of time that could be between weeks and months. And third, the species uses the same area repeatedly for the same purpose of giving birth and developing the young in the first few weeks or months, year after year. So these three criteria are what makes possible for us to define if a coastal lagoon is a nursery area for sharks or not. And now I'm going to talk to you about what we're doing in Mexico. The project that we're dealing with is called Future ISRS, Evaluation of Shark and Ray Nursery Areas in the Mexican Caribbean. And we're studying two different coastal sites. Bahía de la Ascensión, which is a protected area since 1987, and Laguna Yalajao, which is kind of protected since 1994, but I will explain you what I mean by kind of protected. This is the coast of Quintana Roo in Mexico, where I live, is in the south. And these are the two bodies that we're studying. Bahía de la Ascensión is this one in the middle of the coast, and in the north is Laguna Yalajao. Now, Bahía de la Ascensión, as I mentioned, has been protected. A ban on fishing nets was established almost 40 years ago. So there have been no nets of fishing nets in this area for almost 40 years. And that has been fantastic and is very beneficial for all the fauna that lives there. However, in Laguna Yalajao, they have still allowed for fishers to still use nets inside the lagoon. And this is very, very detrimental, and we're going to see how that plays a very important role in what's happening. Now, the objectives of our project is to identify areas of special importance for conservation of sharks and rays in the Mexican Caribbean, and use that information to feed into the ISRS identification process, particularly in nursery areas. And we want to determine in each of our study sites if they meet the three criteria that makes shark and ray nursery area by the definition of these other scientists that I mentioned. So we're looking at those two places, and we're trying to determine for which species of sharks and rays this applies, or this is the case. Now, our research plan includes traditional and modern techniques to investigate if the three criteria are met and for which species. Shark and ray species that still use each site as a nursery, and the seasonality of their use of those nursery areas. And we also want to find out a little bit about the partitioning of each body of water by different species, because we don't expect that they will occur one on top of the other, but they're gonna kind of divide. You go to that side and we go to another side, and we use the habitat differently for the same purpose. So that's the main part of our research. The methods we're using, first of all, is traditional fishing gear. We're using long lines that we monitor every 30 minutes. We're using also something called broops, which are baited remote underwater video systems. Basically, a camera that is put to record in front of bait on the water. We are also using drones to do aerial transects and see if because these are very shallow waters, if from there we can see what's happening, what species occur there. And we're also using a very new technique, very powerful, called environmental DNA. Now, long lines for small sharks and rays are 50 hooks of the size 12-0 for the hook. Is not a very large hook because we try to catch smaller sharks and rays. The main line is made of 300 meters of threaded nylon. And this is a picture showing you our type of gear. It's basically a very long line that has vertical lines with hooks on each of them. So it's 50 hooks per set of the long line. And there are ganglions, which are the vertical lines that are made of nylon and wire, so they cannot be broken easily. And there's a buoy every 10 hooks. We monitor the long line every 30 minutes to make sure that the sharks and rays do not die. Because if you leave a shark in a hook for a very long time, it's gonna die. And we don't want them to die, we want to keep them alive. So we put the long line, and then 30 minutes later, we go and check it. And then an hour later, we take it out of the water and move to another place. Broops are structures made with PVC. Here you have a photograph of the model we chose. We took it from the literature, and there's a GoPro camera attached to that to record. The bases or skates are filled with concrete so that they weight down and keep the structure in place in the bottom. We put concrete on them, and the fish bait is inside a capsule that is here in the front, that is made of PVC and has a lot of holes so that the flavor, the smell, the oils, the blood can come out and attract. It serves an attractant for sharks and rays. Each broob is placed for 90 minutes, an hour and a half, to record video, and then we analyze that in the lab. Now, the design of sampling in each place is such as this. We put a lot of effort every time we go out to each. This is Bahía de la Ascensión, and these are the extensions for long line. We put 32 long lines every time we go out there. Takes about 10 days of work, and we put 72 broops for an hour and a half at the same time. So we work in teams in 2 boats, a lot of money, a lot of effort is spent into this. The other side, we have a similar approach, but it's a smaller side. So we use less amount of days. We only sample this in 5 days, and we are putting 20 long lines and I believe around 40 broops. 40 broops in Laguna Yalajao, which is the other side in the north. Now, to assess the diversity of sharks and rays using eDNA, we are using this technique that is great to detect species even when they are at very low densities. If you only have, in theory, only one shark living in a coastal lagoon, with the DNA, we can detect it if we're lucky, even if we don't see it and we don't have to handle it or record it with video or anything. We just take a sample from the water and we can determine if that species exists there. So no contact or direct recording of the organism. And this is based on finding DNA in environmental samples that can be water, can be sediments, or even in the air. And this is a very powerful technique that it has already been proven to work with many species of aquatic animals, including sharks and rays. To be a little bit more in detail into eDNA, it is the DNA, environmental DNA technique is based on all the DNA, all the cells, all the tissue that we as organisms and fish and seals and dogs and cats and everybody lives in the environment. In the marine environment, fish can leave scales, slime, feces, urine, gametes, their dead bodies or injured bodies in there, and they are releasing constantly cells and DNA. So we take that DNA, we extract it from the water samples, and then we use that to detect whether that species is present or absent in a certain area of the sea. This depends a lot on the persistence of DNA in the water. DNA degrades, bacteria, ultraviolet light, temperature, break it down to atoms and molecules that are no longer distinguishable as DNA. So there's only a relative short time of that, and that depends on the density of the species, how many organisms live in a water body, and also on the environmental factors such as temperature, ultraviolet ray. This little cartoon that you see here shows time in red, not very good choice of colors, but you see a fish swimming there, and there all those traces of DNA that is left behind. As time passes, that DNA disappears. And this is the magic, not magic, but the great principle of environmental DNA. An organism is constantly losing DNA into the environment, but after a short while, would be days or weeks, no more than months, that DNA is going to disappear. So if I go to a river or to the beach and take a sample, and I find the DNA of a white shark, I know that a white shark was passing through that water or nearby in no more than 2 or 3 weeks, because after that, that DNA would not be recognizable. So that's the important principle. So the samples we take are water samples of 1 liter, and we take them from a boat using a special pump. And here you see me with the hose. And this, in my right hand, I have a capsule with a filter, a special filter inside the water, and it's pumping water into this bottle here that collects 1 liter of water. And that's what we do. After that, we are using a very small membrane filter that has 5 microns of mesh size. So it captures very, very, very tiny particles. And that's the capsule with the filter here. I'm taking the filter out, and then we keep it in silica gel and freeze it immediately that night and keep it in the lab. We send it to a lab for processing. The processing of the eDNA samples in the lab implies the extraction with special chemicals of the DNA from the filters, and then running something that is called a primers. Primers are molecules that cut specific parts of the DNA, and that's what we use to find parts of the DNA that will tell us what species of shark or ray we are capturing. The technique normally is very simple when you're dealing with one species, and you can do it with something called gel electrophoresis. But in this case, we're trying to find all the sharks and all the rays, and that implies buying getting access to a very expensive modern technique called a Illumina sequencer. And this automatically does the sequencing of the samples and processes them and gives us the information. Now, we only need to take about 9 samples of water, 9 liters of water, well spaced around Bahía de la Ascensión for the eDNA. And in the other place, we only take about 6 samples. So only 6 liters in one place, 9 liters of water every time we go. And with that, we are set to do our eDNA. We also take parameters with a special parameter meter. We measure the temperature, the salinity, and the dissolved oxygen in every station where we're sampling to be able to correlate the environmental factors with what is the abundance of each of the species. And now I'm going to show you a bit of how we do the work. We're setting the long line here. It's a careful process, and then we have to check it after 30 minutes. And in these photographs, we are taking a shark in one and a ray in the other after 30 minutes of checking. Every organism that we capture, we have to measure it very carefully in the length for the sharks, the disc width for the rays. We also insert a plastic tag to identify each individual into the back of the sharks of the rays. And in these two photographs, you can see my students applying that small plastic tag. And here is one, and that yellow thing, tiny little yellow thing that you see here, like a spaghetti, that's the tag. And it has numbers and letters with a message there. So if a fisher catches, they can report to us that number and where they caught it, and that is very useful information for us. We always remove the hooks from all the organisms that are caught in our long lines to ensure that when we return them to the water, they can live normally and survive. We also take small clips of fin of the organisms to do other genetic studies about populations. And sometimes we have to keep, when we have many sharks or rays in a single set of the long line, we have to babysit them for a while somebody's working with the tagging and measuring of one on the boat. Somebody goes with going with your dog on a leash. And here one of my students is with a small baby lemon shark, keeping it alive and moving around so it doesn't die while we finish the work on the boat. And these are a few other images of a bull shark, a sizable juvenile of about 150, 160 meters, really big round ray. And here we are returning that bull shark back to the water alive. And then we try to release of our organisms alive. Sometimes when we go and check the long line, there's already an organism that's dead. We don't like that, and we try to minimize it, but occasionally it happens. So this is gonna be a little video showing you how we check. This is the boat. We are checking the long line. You can see the main line there. And we're pulling that main long line. We're passing a buoy now, and vertically each 3 meters approximately, there's a ganglion with the hook. And in this very small video, there's not gonna be any sharks and rays caught, but I decided to include it just to show you the area where we work in Bahía de la Ascensión and how we do the work. Okay, and now this is a bit more interesting, and this is how we one of my students and myself dealing with a
67, 67.
Measure it.
Macho.
Check the sex.
Cuidado.
Okay, a male. We have to tag it. Sometimes not as easy as you would like. They have very rough skin and very difficult to cut through.
Ya está.
And that's the tag being inserted. It's a spaghetti tag. There it is. Already secured. We're almost done. But in this case, I'm taking a film clip from the anal clip, a bit of tissue. I'm telling my student to return it to the water because we're not done, but it's been too many minutes. The shark out of the water.
Más falta cortar el suelo.
Let it breathe.
Ya.
And then we
Y checar la cicatriz.
that they don't like. This has proven very successful. We have very few deaths. And now just the final thing, take the hook out. Got it? Got it right. And now as soon as it's returned, there it goes. Back to safety, back to its environment. So that's how we do all the work. Now, the broods, these are some of my other students preparing a broob on the boat. This is the capsule with the bait, and I'm filling it with dead fish to attract the sharks, and then we just set it on the bottom And this is in a very shallow part of the lagoon. The group already sat there, started to record, and we leave it alone for an hour and a half. We go away so sharks and rays can come, attracted by the bait, and then we will see what species occur in that particular part of the bay. A few more images of the setting of the groups. And this is a group already in a shallow water. It's an underwater photograph. And this is in the deeper part of the reef. This is in about 12 meters of water, or maybe 10 meters of depth. And these are already recording whatever happens. And here are a few of the results of the groups, some shots that I took from the video. This is a baby blacktip shark. And this bar that we put here helps us measure the size of the shark so we can determine how large it is and find it is a newborn or it's already a bit longer. Maybe it's been there for 6 months or a few more months. This is a Caribbean reef shark. Here we have another shot of the same Caribbean reef shark that is very, very curious, trying to get the bait. The typical nurse sharks are a nuisance. They're always coming to the groups, and we cannot tag them because they have extremely thick skin, and we cannot take fin clips of them. So basically, we only measure them and return them to the water. And then this one shows there a lemon shark, a baby lemon shark. They are very shy. They don't come as close to the bait as the other sharks. You can see here, this is keeping well away from our group, and the other sharks are really trying to get to the bait. And this allows us to see the different behaviors of each species, and we are already detecting that lemon sharks are very different in behavior from the other species of sharks. And here's a little bit of a video of the work with the drops. I think we're running a little bit, taking a little bit too long with this. I'm gonna try to speed this up to present to you the results and things. And that's a Caribbean reef shark. That's an Atlantic chicory, a ray, a down ray. But no suppose, even a toro. It's not in orange, but I thought I like it. And that's near lemon shark. How the lemon shark never come too close to another ray. And nurseries. Another one. And then we take the food out of the water. No. All right. So
That's really amazing, that footage.
Okay, I'm glad that you like it.
Yeah, that's really cool.
We're almost done. Now, the results, the preliminary results, long-line sampling in Bahía de la Ascensión, we already caught 230 sharks and rays. This is in the first trip, 104, second trip, 69, and in the third trip, 57. Every trip since we began in September, the abundance is decreasing and the catches are decreasing, which suggests to us that we started right when the babies had been born, maybe a month or two before. And as time passes, they are leaving the area. We cannot say that for sure yet, but we have great information, a lot of species, a lot of sharks and rays and good numbers, and we managed to tag a lot of them. And an interesting thing, our mortality in the first trip was 11 out of 104, about 11%. Next time it was less than 2%, 1.4%. So we decreased the mortality substantially. And on the third one, we kept it not as low, but still good, 5%. Now in Laguna Yalajao, we have only got 50 sharks and rays in the 3 trips, 11 in the first, 27 in the third, second and 16 on the third. So the numbers are completely different, and the amount of species is also very, very small. The diversity is smaller. And these are a few of the preliminary results of the distribution of shark species and race in Bahía de la Ascensión. The lighter colors toward the red and green and yellow is where more abundance, and the darker blue means no sharks at all. So this is for the blacktip shark, and this is the center of abundance, but you can find them all over the place. This is very different. This is for the lemon shark, and it shows us that they occur in very different areas. They split the Bahía de la Ascensión. You go there and I keep there. So it's a good partitioning. This is for the round stingray. It's all over the place, but not very, very, very abundant. It's relatively abundant, but not too much. And this is the sharp-nosed shark that is mainly in the mouth of the bay towards the sea. Doesn't enter very deeply into the coastal bay. Now, in the longline sampling in Bahía de la Ascensión, when we compare it to Laguna Yalajao, it shows that in Bahía de la Ascensión, we have a total of 230 individuals caught, 7 species of sharks, 3 with early juveniles, 2 species of rays, and none of them with juveniles. While in Laguna Yalajao, the other side, we only have 54 sharks and rays. Of course, we have less stations. We spend only 5 days as opposed to 9 days working. So obviously we're gonna have less catches, but the proportion is too large. We should be getting more, about half of the sharks and rays in one place than in the other, and we're getting less than a quarter. 3 shark species only detected, 1 with a newborn, but it was not inside the lagoon. It was in one of the stations that is outside of the lagoon, and 3 ray species. So none of them with early juveniles. So you can see the contrast in diversity, and the abundance of sharks and rays is very, very contrasting between the protected, the really protected area where no fishing nets are allowed, and the other place. Now, from the groups, we have 4 species of sharks in Bahía de la Ascensión, 2 with early juveniles, 2 ray species detected, none with any juveniles. And here is a graph showing you that the most abundant species we've found in the groups in Bahía de la Ascensión are the nurse sharks. In Laguna Yalajao, we haven't finished all the analysis of the videos, because it's a lot of hours on screen checking minute by minute what shows up. We have 3 species of ray and 1 species of shark found. So our conclusions so far, which are preliminary, I stress preliminary, is that there's a notable less diversity of sharks in long lines in Laguna Yalajao. We have 7 species in Bahía de la Ascensión, but only 3 species in Laguna Yalajao. We have lower abundances of sharks and rays in Laguna Yalajao, 2.12 sharks and rays per set in Bahía de la Ascensión, and less than 1 organism per set in Laguna Yalajao. And only 5 sharks have been caught in Laguna Yalajao versus 195 in Bahía de la Ascensión. So most of what we get in Laguna Yalajao are rays. There's less diversity of sharks in Laguna Yalajao, according to the groups also, 4 species in Bahía de la Ascensión and only 1 species in Laguna Yalajao. And there's a lower relative abundance of sharks in Laguna Yalajao, according to groups. So it seems so far that Laguna Yalajao might no longer be a nursery area for shark and rays. 20 years ago, somebody else did a study, and they found that it was a very important nursery area for blacktip sharks. We haven't found a single blacktip shark in almost a year. So it seems like Laguna Yalajao has lost its function as a shark nursery area, at least for that species. And it seems so far that Bahía de la Ascensión is likely to be an important nursery area for several sharks and maybe even a few rays. But we still have to finish our sampling. We're about to finish by the end of the year, and then we're gonna have to analyze all the information we have. So with that, I wanna thank all of your time and acknowledge all the students and volunteers that have helped us, all the fishers that have helped us with the work in both places, and also the Mexican authorities that are giving us the permits that are required to do research in national protected areas. And with that, I return the microphone to Lance.
Yeah.
I'm happy if there's still time to answer a few questions.
Yeah, well, I think we have some time for a few questions. That's amazing. Yeah, I mean, I appreciate as a ecology student way back when, I appreciated your ecology lesson early on, and explaining why sharks have this issue, right? Of low reproductive capacity, and how taking so many out so quickly is causing so many problems, right? Sharks have adapted for millions of years to be the apex predator. Ecosystems need low amounts of those apex predators, otherwise they throw the ecosystem out of balance. So, but they're haven't adapted to humans taking them out so fast. But so I appreciate that first in the first yeah, that was nice. I just had a few questions come in while you were presenting. You mentioned the CITES listings. What does it take to get a species listed on CITES to have that international protection?
It's not easy. I had the fortune of leading an effort 21 years ago, 22 years ago almost, to propose that the white shark was listed in Appendix 2 of CITES. And first of all, you have to write a proposal. You have to write a very detailed document that demonstrates that this species needs protection, because you have to state all the reasons, and there's a number of things that you have to comply with, requirements that you need to comply with. So it involves a team of scientists putting this together, doing this document. Is a literature study. Takes months, and then once you have it ready, you have to find a country or a few countries that are willing to propose it, to submit it, because me as Ramón Bonfil or as a scientist, I cannot do it. It is the governments of the countries that sit down around a table to discuss these things. So we find a delegate from 2 countries. In that case, I think it was the Philippines and Australia, and they together took what we scientists prepared and they submitted it on behalf of their governments. And then it was discussed, and there's a lot of analysis, and there's a lot of politics going, economics and politics going on at play in these meetings, because many countries that have interests in fishing, they don't want any restrictions with their fishing activities. They want to keep fishing without being disturbed. But the conservationists, we want to protect the species. So there's always a clash, and there's a lot of people trying to convince others to. Some governments try to buy votes in other governments by saying, oh, we'll give you, I don't know, an airport, or we'll give you roads, or we'll give you millions in help, in aid. And then somehow they succeed. But we started, I think the first shark that was listed was either the basking shark or the whale shark, the first ever. And that was in 2001. And then it was the other one, the basking, and the third one was the white shark. But after that, now there's over 100 species of sharks and rays in Appendix of CITES. So things are gaining momentum, but still it's not enough. I mean, the rate of exploitation keeps increasing. The amount of people wanting to either fish or eat fish keeps growing, and we're running over the world, the planet, unfortunately.
Well, I mean, it speaks to how difficult it is to get something listed at such what feed it is. So, you know, for what we do have, congratulations for what you have been able to help us with.
Thank
Um,
you.
with your current research, I think we have time for maybe like two more questions. With
Okay.
your current research, I know you so with the shark nurseries and you have preliminary results. How would you expand on that? Do you have any plans to take the next step? What would that be to
Yeah,
expand what you're
well,
doing?
there's two options. One is to go to other bays. We have two other bays, large bays
Mm-hmm.
in the coast of Quintana Roo that I would like to find out if they are nurseries for sharks and rays and for which species they are. So it would be reproducing the same study in other nearby areas.
Sure.
But that would require a lot of money and a lot of people involved. At the moment, I originally planned for 8 students to be part of this project. I had to adapt it, and now we're only about 4 students or
Mm-hmm.
5 doing this, and me and myself, a lot of work.
Yeah.
The other thing is in Bahía de la Ascensión, we found this specific area in the south part where lemon sharks are very abundant and well known to occur. And we want to study that more in detail and see if we can catch a mother giving birth, just like the videos from Bimini that I show you. I don't think that has been documented in many parts of the world, maybe only in Bimini or maybe in 2 places. I would like that to be another place where I think this is an ideal place because it's
Right.
very low population of humans, a lot of lemon sharks there. So we need to be lucky, get the money, not that much money, but a little bit of money, and spend there the right amount of time exactly in the moment when the mothers enter to be
Right.
able to film and document babies being born there. And that would be wonderful.
Yeah. Well, Save Our Seas watches these videos. You never know. um, well, the last question is just if anyone, you know, people that are watching, how could they stay in touch? How can they learn more about what you're doing? Is there anywhere they can stay connected?
I forgot that. Well, yeah, we have a maybe if I write here on the chat,
Yeah, oh yeah, it'll come up.
I'll leave you my email here. Well, you have it, but that's where they can write to me. And another, we have uh, a little bit of
Okay. Oh yeah, and you have the website EcoSur, right?
Facebook and
.mx.
Instagram. We have something in Facebook and Instagram, but I never remember what it's
That's
called.
okay.
But I'll find out and I'll send you the
Yeah,
information.
I've perused the EcoSur website a little bit, and you can find a lot of scientific literature on that page as well, and
Okay.
learn more about you and everything too. So that might be a good option for people. And you posted in the chat, so thank you. And your email. So if anyone has any questions, feel free. Ramón.Bonfil at ecosur.mx. Well,
Okay,
Doctor,
found it.
thank you. Oh, yeah.
I'm gonna just paste here the website in Facebook.
Oh, okay, great.
How that
Oh,
works?
there we go.
It's called EcoSharks.
Eco
And
Sharks on
I
Facebook.
hope with
Okay,
that
great.
one you can see the activities that we do there.
Okay.
Some posts that we have of our work.
Fantastic. Well,
All
thank
right.
you very much for coming on
Well,
and
thanks to you, Lance.
explaining your work.
It was a pleasure to share my work and my knowledge with all these very interesting people that are keen on learning more about the sea and conservation.
Yeah. Well, we certainly appreciate it. You coming in and sharing such a holistic project, and then all the giving us an ecology lesson all at the same time. I really appreciate that.
Okay.
Join us next time. We're going to be having another speaker come in on July 24th, the next iteration of the Save Our Seas Foundation Distinguished Speaker Series, Deep Water Sharks of the Caribbean, with Ana Hernandez from the Blue World Foundation. So July 24th, 6 to 7 p.m. You can go to mods.org/STEM Learning Series to sign up for it, or scan that little QR code there and join us. We'd love to have you. Thanks again to the Save Our Seas Foundation for making this whole series possible and dedicating so much to the world of shark and ray conservation and education research. And thank you to all of you for being here tonight and listening in. Hope everyone has a great evening, and we'll see you next time. Bye.
Bye.